associates with ribosomes. Okay? Where do we then, how do we differentiate in the process? If we're going to synthesize, enzymes that go to the mitochondria. What organelles are those ribosomes and messenger RNA associated with? The ribosomal ER and the Golgi. So the ribosome and messenger RNA complex is attached to the ribosomal ER, and the new proteins are inserted into the cisternae of the ribosomal ER, and then are pinched off in a sac and transported to the Golgi. Within that membranous sac, they have a destination. So part of the new protein is inserted into the membrane and says this sac of proteins is destined for the surface. It's digestive enzymes. It should be secreted from the apical surface. All right? Or it's hormones. It should be secreted from the basal surface. Or it's mitochondrial enzymes. It should be sent to the mitochondria. Or lysosomal enzymes. So anything that has a designated destination is going to be packaged via ribosomal ER to Golgi. Our enzymes in the cytoplasm do not have a designated site to go to. So those ribosomes are free ribosomes. They form the polyribosomal complexes. And as the amino acids are strung together and then they reach the end of the messenger RNA, they're just released out into the cytoplasm. They have no designated route. As I said, each of these chemical reactions has a specific enzyme for that reaction. And all of these reactions, except the first one, are reversible. So the addition of this first glucose molecule forming glucose 1-phosphate is non-reversible in our cells except in the liver. Because our liver is going to take glucose, phosphorylate it, and go through a series of reactions to store it as glycogen. And then when our body needs glucose, all of those reactions are reversed and glucose is released into the bloodstream. So that first step of adding phosphate is reversible in the liver, but not typically in our other cells. Okay, but all the other reactions we're talking about are reversible and are driven by concentration components. Okay, so before we can start these reactions and release the energy down in the cell, let's say that we want to squash a bug. It could be a ladybug or that could be a Volkswagen bug. And we've got this huge rock that we're going to push over the hill. But to keep that rock from just randomly rolling over the hill when there may not be a bug down there to squash, there might be your foot, there's a lip to keep that energy located where it is. And so to get that rock and that potential energy to fall down here, we have to add additional force to push it over that lip, that activation energy. And so that's what we're doing in the first step here. Before we even form energy, we're adding an activation energy to our glucose molecule. So that's where these first two ATP molecules come from. So we start out, we're going to utilize glucose here, although we could start with fructose. So we're going to take a 6-carbon. I could count this morning and I had 7 carbons up there for a while. We're going to take our first molecule here, which is glucose, and we're going to add ATP, and I'm going to draw a one-directional arrow because this is not reversible in most of our cells. We have a phosphate group. So we form then the glucose is rearranged and for glucose 6-phosphate, the glucose is rearranged to fructose. So instead of a 6-sided ring, instead of a 6-sided ring, we have a 5-sided ring, and so the name is changed now to fructose. molecule, and each of these is regulated by an enzyme. Okay, you don't need to know the specific enzymes. In fact, the specific components of this material that you need to know is given at the end of each series of reactions as a summary. So you'll see a summary in the middle of page 56, and that's everything you need to know about glycolysis. All right, and then you'll see a summary at the bottom of the page, and that's everything I want you to know about the intermediate step. And we'll see those as we go through this material. Again, you don't need to know these individual enzymes. And my son had to learn them for high school biology. I had a roommate in graduate school that was in med school at the time, and our entire bathroom wall and our shower curtain were covered with biochemical reactions and all the enzymes. So I'm not asking you to go home and do that, all right? So just what's on those summaries. So we add another phosphate group. So now we have fructose 1,6. This is the international nomenclature. So anybody reading this would know where to put the molecules. And along with this is our activation energy. So it's taken up 2 molecules of ATP. We're already in the hole for 2 molecules of ATP. But now we've pushed that rock to the very edge of the cliff, and we're teetering. We're ready for that potential energy to be made available. So the next step now is breaking our first carbon-carbon bond. Notice we don't get any ATP from that. Okay, there's not enough energy released in breaking that carbon-carbon bond to store as ATP. In fact, most of the energy we get is in the hydrogens. Okay, as you'll see by the time we get to the very, and electrons of the hydrogens. All right, one more reaction, and I'm going to erase this and start at the top of the board again so you guys can see it. So we take our fructose 1,6 diphosphate and break that bond, and now we have 2 3-carbon molecules. What 3-carbon molecule did we talk about yesterday? Glycerol. So this name is similar. Each one has just 1 phosphate, and this is called glyceraldehyde 3-phosphate. Now remember, there's oxygen and hydrogens on here. I just haven't drawn them in there yet. And we have 2 of these. So this is very similar molecularly to glycerol. So when the body switches to using fats for energy, and they take triglycerides and break them down into fatty acids and monoglycerol or glycerol, this is one place where those components of the triglyceride can come into and become, utilize this reaction. So over here we have fatty acids and fats coming in. So glycerol can come in here at the glyceraldehyde stage, and the body doesn't know the difference, and it can pass through and be used as a fuel source, right, because of its similarity to glyceraldehyde. So we're at this step right here. So we've taken glyceraldehyde 3-phosphate and at this point, and now we're going to remove for the first time some hydrogen molecules. Okay? We still have 3 carbons when we're done, but it's a different molecule. It's pyruvic acid instead of hydrogen. At the same time, when we remove those hydrogen molecules and atoms, sufficient energy is released to make 4 molecules of ATP. So we've enough to pay back the 2 that we originally used. All right? So I'm going to erase what I have here again and start at the top. Because so far yet, we've just consumed energy. So we have 2 again, enzymatically, in a reversible reaction, we remove hydrogens and we remove our phosphates, and energy is released. We are left with a 3-carbon molecule. Obviously, we now have a few more double bonds because we've taken off some of the hydrogen. So I'm not indicating that in there, but it is a different molecule that is now known as pyruvic acid. And we have 2 of those. So we have, we're doing each of these twice. The hydrogen we're going to see again. It's going to be our source for most of the ATP that is formed in the cell. So of 38 molecules, 34 of those molecules of ATP are going to be derived from the energy in the hydrogen. Okay? As we go through our last stage, our electron transport, down here, way down here. So we have to move this hydrogen, we have to send it to another side of reaction, and to do so we use a coenzyme. And that coenzyme is NAD, nicotinamide, and so we end up with NAD. It's written a couple of different ways. You can see it like this, or written like that. The phosphates are removed, and along with additional phosphates and ATP and ADP and energy, ATP is made. A total of 4. There's enough energy to add the 3rd phosphate to 4 ADPs. Now, if this was a correctly written chemical reaction, we'd have ADP on this side and the additional phosphates and energy on this side, but I'm more interested in showing you what our products are here. And so we take the hydrogen off of the glyceraldehyde, attach it to our coenzyme. The phosphates and the energy are sufficient to make 4 molecules of ATP. If we subtract the original 2 that we used to prime the reaction, we're left with a net gain of 2. Now, I'm not going to ask you to keep track of how many ATPs come from hydrogens and how many from FAD and how many from each cycle, but I'm just telling you as we move along so you get an idea kind of how this is spread out. Notice that while we have made ATP, this is the first time that we have ATP from our glucose molecule, we have yet to release any carbons. So we still have all 6 carbons. We've removed hydrogen and we've captured energy, but we still have all 6 carbons. All right? And that is the final product in an aerobic state of glycolysis. Now, glycolysis itself is considered to be anaerobic. It doesn't require oxygen to be present in the cell. But if we don't have oxygen present in the cell and we go through glycolysis, our pyruvic acid is converted to lactic acid. Okay. So pyruvic acid is our product of glycolysis occurring in an aerobic environment. Again, without oxygen, its next stage would be the lactic acid. So if you look on page 56 then, under summary, okay, I've listed the events and the results that I want you to be able to recognize as occurring with glycolysis. So what are our products? ATP, NADH2, and the pyruvic acid, the 3-carbon molecule. Okay. Now, before we can consider the reactions that take place in the mitochondria, we have an intermediate step. It's a 2-carbon molecule. So we have the intermediate step, which it takes place in the cytosol, and that's now the first time. This is the first time we formed ATP. But in our intermediate step, up at the very top of the screen, is the first time that we release carbon as carbon dioxide. So we're going to go from a 3-carbon molecule to a 2-carbon molecule. So the next event, this is the end of glycolysis. The next event is the intermediate step. No, prior to the intermediate, prior to the citric acid cycle. We're making the precursor component that goes into that. So in our intermediate step, we have 2, again, molecules of pyruvic acid. And this will only go forward if the cell is aerobic and has oxygen available. And now for the first time, we're going to lose carbon as carbon dioxide. As that happens, hydrogens are also released, like we saw before. Those will be picked up by our coenzyme. And since we lost the carbon, our 3-carbon molecule is now a 2-carbon molecule. Again, it's still going to have hydrogen and oxygen as a component of that molecule, simplifying it. And this is our acetyl group. This happens twice, one for each molecule of pyruvic acid. Remember, we started out with 6 carbons? So we still have 6 carbons here because we have 2 molecules of pyruvic acid. This happens twice. So we make 2 molecules of carbon dioxide, and we're left with 2 acetyl groups. So you can track the carbons. 1, 2, 3, 4, 5, 6. Our coenzymes are going to transport the hydrogen to the mitochondria, and our acetyl group also picks up a coenzyme. Coenzyme A, so we have a molecule called acetyl-CoA, and that's what starts our citric acid cycle. Yeah. Yeah. So this now is what we take to the mitochondria and start with our citric acid cycle. So the mitochondria is an organelle that has 2 membranes, an outer membrane and an inner membrane known as the crista. You remember that from Bio430? And it creates several environments. So we have the inner matrix. This is where our acetyl-CoA is going to end up. And we have the outer matrix. And then we have the cristae. And we have the outer membrane, which we're not going to talk about. So we're going to talk about the inner matrix, the cristae, and the outer matrix as we talk about the citric acid cycle and electron transport. So if you look at the diagram up here, there's the acetyl-CoA, the 2-carbon molecule. All right? The CO2 is diffusing out of the cell. And this is now within the mitochondria in the inner matrix. And that's where the enzymes are found that regulate these series of reactions. They're not located in the mitochondria like a fairy ring. Okay, that's not why this is called a cycle. It's called a cycle because the end product of all these series of reactions is one of our substrates to begin with. So we end up with what we started as far as the oxaloacetic acid is. So we're going to combine oxaloacetic acid with our acetyl group, go through a whole series of reactions, and we're going to end up with oxaloacetic acid again. All right? That's why it's called a cycle. These enzymes can be located anywhere within the inner matrix. And it's purely random where each of these reactions take place. So these blue dots represent the different enzymes regulating each of these steps. And so the first reaction can occur here, the next reaction over here, the next reaction over here. So it's not going around a Ferris wheel. Okay? It's just a cycle because of our final product was one of our first substrates. So the molecules that I write on the board are the ones that I want you to know. There's only 3 of them. So we're going to come back to our acetyl group. The coenzyme A dissociates and goes back out to pick up another acetyl group. This is going to bind with a 4-carbon molecule known as oxaloacetic acid. Those two form a 6-carbon molecule. We're back to the 6 carbons we've been trying to get rid of. But it doesn't have the same number of oxygens. All right, we've already lost 1 oxygen from each acetyl group, and we've lost hydrogens. So it's not the same molecule, and this is known as citric acid. Hence the more modern name for this process. You can see that a variety of reactions occur as we go through to get back to oxaloacetic acid. I'm not going to draw the different products, secondary products that are formed here, but what is given off, I will diagram. But the point I want to make here is this is a site where other additional amino acids can come into and be used for fuel. So different amino acids can come into these different sites and be incorporated in the citric acid cycle as new sources of fuel instead of glucose. All right? So different hormones will increase. Someone that has a high quantity of cortisol will start to break down proteins, amino acids. They'll lose muscle mass, especially in their lower limbs, as they're using the proteins for fuel. And those amino acids that are being broken down are being sent into the citric acid cycle. All right, so along the way, and I'm just going to draw one long arrow here, we remove at two different times carbon dioxide. So that's a product of the citric acid cycle. We added 2 carbons, we removed 2 carbons. They are not the same carbons if we were to attach a radioactive tag. You would see that we have to go through the cycle multiple times to get rid of those actual 2 carbons that we added. We will eventually, but not the first time through. Okay? But the math comes out that we added 2, we lose 2. So by the time we're done with the citric acid cycle, all the carbons of our glucose molecule have been accounted for as carbon dioxide. So we brought 6 carbons into the cell as glucose. And by the time we're done with the citric acid cycle and our intermediate step, 6 molecules of carbon dioxide have been formed and diffused out of the cell. So we're left with just the hydrogens to deal with. We've gotten rid of the carbons and the oxygens. All right? So when we move into electron transport, we're going to be dealing with just the hydrogens of our glucose molecule. So some of those hydrogens are also released. 3 times the hydrogens are still, again, added to a coenzyme, the nicotinamide, 1 time to FAD, okay, to a flavin molecule. But it's still a coenzyme. So 4 times, hydrogens are removed from our processing of these carbon molecules and are transported to or by a coenzyme. And again, this is going to occur for each of our 2 acetyl groups. So 3 NADs and 1 FAD, but all of these are coenzymes. So we've lost carbons, we've lost hydrogens, and as we change these molecules, we have sufficient energy. What I've written is just this here and that there. You can't see on the other side. And in addition, we've released enough energy to form a molecule of GTP, which is equivalent in energy level to ATP, guanine instead of adenine. So that's what's shown here on the screen. Okay. So GDP, using energy as a phosphate group, and forms GTP. So remember, we're doing this twice. We have two carbon acetyl groups. Each one is going through this cycle. It may be at the same time. It doesn't have to sit there and wait in line to get on the Ferris wheel, okay? There's plenty of enzymes around. And in fact, that is an advantage to having these reactions take place in the mitochondria. If we had these enzymes out in the cytoplasm of the cell, it would be less likely to randomly, the substrates with oxaloacetic acid and acetyl group would have to come together along with the enzyme. Would have to have a higher concentration of enzymes to make that more likely. So by capturing that within the mitochondria, we increase the likelihood of it's happening with a lower level of enzymes. Plus the products, the hydrogen that we're capturing on the coenzyme don't have very far to go. We're going to send them to the inner membrane, the cristae. So it decreases the time all this takes because the likelihood of it coming in contact is going to be there. So now by the time we're done with the citric acid cycle, we have no more carbon to process. Okay, we've made 6 molecules of carbon dioxide. We've made a little bit of ATP. So we had 2 molecules of ATP from glycolysis, and the equivalent of 2 molecules of ATP by the time each of our acetyl groups goes through the citric acid cycle. So what would be a final product of citric acid cycle? GTP. Carbon dioxide. Our coenzymes carrying hydrogen, so both the NAD and the FAD. And then finally, the oxaloacetic acid, the same thing we started off with. All right. So now we're ready to move on to electron transport. Electron transport is the movement, at first it's the hydrogen, it's being transported from one coenzyme to another, from NAD to FAD. And then the hydrogen is split to form a hydrogen ion and an electron. And from that point on, we're transporting electrons from one component within our cristae to another. And the hydrogen is being pumped into the outer matrix. So that's the process that we're going to look at. A couple of terms to review or to learn, and that's the oxidation reduction. If we talk about in our series of reactions, there are proteins within the membrane that have a pigment in them, and so they're called cytochromes. And they have an attraction. Remember we used the term affinity? So they have an attraction for the hydrogen or for the electron. And that electron is at a certain energy level. The next cytochrome that attracts it away is the electron is at a lower energy level, but it is more attracted chemically to that cytochrome. I'll tell you a little story to help illustrate that in a moment. But before we do so, let's look at the oxidation reduction. So let's say that our first cytochrome is cytochrome A. Without the electron, this protein demonstrates two positive charges. It receives an electron from cytochrome B, and that neutralizes one of the charges and changes it to one positive charge. That is reduction. Okay? So the addition, yes? This cytochrome, the electron is coming from, so I kind of jumped in the middle here. So we'll come back to the beginning in just a moment. I'm just illustrating the definition. Okay? But let's say it came from cytochrome C or from cytochrome B. And this cytochrome has a higher affinity for it, so it draws it away. And it neutralizes, balances the positive charge, cancels out the positive charge. So when that happens, this is reduction. But then the next molecule, lower down, but with an energy level-wise, but with a higher affinity, a higher attraction for that electron, pulls that electron away. This had two positive charges. That electron is pulled away now, and will reduce this molecule, while this one goes back to two positive charges. When that happens, this molecule has been oxidized. All right? So the way I remember it is reduction is not the loss of the electron, but reducing the number of positive charges. So when we add an electron, we reduce the positive charges by one. And when we take the electron away, oxygen, remember, needs electrons. When we take the electron away, it's been oxidized. In chemistry, you may have been introduced to this phrase to help you remember. Oil rig. Oxidation is loss of an electron. Rig, reduction is gain of an electron. Okay? So whichever helps you figure that out. So that's what we have in electron transport, is a series of oxidation reduction reactions. So it's not really an electron, but when we add the hydrogen to the coenzyme NAD, we are reducing it. And then when we take that hydrogen away and add it to the coenzyme FAD, the NAD has been oxidized. Okay? It's not quite, we're doing with hydrogens rather than electrons. So what we're going to follow then is each of those subsequent molecules, the cytochromes, ubiquinone, that after the FAD, the hydrogen is split to a hydrogen ion, and subsequently we're just transporting electrons, hence the term electron transport. About 3 times along this pathway, there's sufficient energy difference in the energy required by the cytochrome downstream to keep that electron, sufficient energy that it doesn't need, that energy is used to pump hydrogen from the inner matrix to the outer matrix against its gradient. All right? So we're using up the energy that's being released to move hydrogen against its gradient. Eventually, as those hydrogen ions increase, they will return to the inner matrix via an inner channel in this molecule, kind of like water at a water turbine plant. Electrical turbine plant is at a river dam, and the force of the water causes the turbines to spin and converts that spinning and the magnetism to electricity. And I'm not a physicist, so that's about as far as I can go through with that. But it causes this ATP synthase to spin, and that energy that is a result of that spinning is used to make finally the ATP. That energy is captured as that third phosphate bond. So that's kind of the whole story there in a nutshell. But I want to go back and illustrate the changing in attraction and energy levels. It's not very flattering to women. But as I told the morning class, since I'm a woman, I guess I can tell the story without too much flashback or
damage. And that is, there's a young woman, or a middle-aged woman, whatever, she's at a bar. That's probably the beginning of her problem. And she starts talking to this guy. And he's just really nice. Somebody she didn't expect to meet there. And, you know, they like the same kind of music, and she just likes his personality. And he thinks she's cute, and she's fun to be with, and he has fun talking to her. And so he invites her to go home with him. And she does. And he's got this 50,000 square foot mansion. And when they drive up in his Jaguar and park it in the garage, there's 2 more Jaguars and 3 Ferraris in the garage. The next morning when she wakes up, the maid brings the orange juice, and there's 3 10-carat diamonds in the bottom of her crystal glass of orange juice. And he says, I've got to go to work, but my helicopter's out in the yard, there's cars in the garage, here's all my platinum credit cards, just do whatever you want. And she feels like she's in heaven. She's got this great guy, and she's got everything she could possibly think of that she would want. He comes home, and they go skiing, and they just have a blast. Well, she ends up back at the bar, because he's off at work one day, and she finds another guy that she's talking to. And, you know, her first friend, they saw the same movies, they liked the same music. This guy is different. And he's seen movies she hasn't seen, and he's read books that she hasn't read, and he's been to countries that she hasn't been to. She goes home with him. Well, his house is only 10,000 square feet. There's 10-carat gems in her orange juice, but they're only rubies. He doesn't have a helicopter. But she's happy, because she sees him a lot. She's attracted more to him. And even though he's not spending as much money on her, she doesn't need that to keep her there, because he's just so much fun to be with. She doesn't know that he has just as much money as the first guy. She doesn't really care, and he's taking all that money he's not spending on her, and he's buying investments and properties and stocks and islands and stuff, making ATP. Well, that lasts a while, and she goes back to the bar. And each time she goes back to the bar, she doesn't realize, she doesn't see this pattern, but each time she goes back to the bar, she finds someone that's more interesting than the guy that she's been with, and therefore they end up spending less on her. Eventually, she finds herself going home with a guy who has beachfront property. He's got a tin shack right on the beach. And outside are 2 surfboards, and they have a blast. She can see herself spending the rest of her life with this guy. No helicopters, no cars, no credit cards. They've got the water and the surfboards and each other, and she's perfectly happy. So each guy had a higher attraction for her, but needed to spend less money to keep her, right? So if I have to spell it out, she would be the electron, okay? The attraction that she had for the guy would be the cytochrome, or the ubiquinone pigments, or the NAD. And that attraction they have for each other would be the chemical affinity. The energy would be the money. The more money he didn't have to spend on her, the more he could use to pump into his investments. Okay? So as she moves from one guy to the next, she is reducing the guy that she is moving in with, and the guy she's left is being oxidized. Okay? Again, each molecule that the electron moves to is at a lower energy level. Finally, we get to the lowest energy level, and we recombine everything, having given up the energy. The hydrogen flows down its gradient, not needing energy to pass through the ATP synthase molecule. It flows down its gradient from the outer matrix to the inner matrix, and as it flows through, this rod spins, and there are enzymatic catalytic locations that enzymatically add phosphate to ADP and create ATP. So the difference in energy between where we started at NAD and where we end up with hydrogen is enough to form 3 molecules of ATP. So our final result here, at our lowest energy level, is an electron at its lowest energy level. Hydrogen ions. So we'd have 2 of these, plus half a molecule of oxygen gas, forming water. That's our final product of both electron transport and glucose catabolism. Okay. So when we're finally done with glucose catabolism, our final products are ATP and water. What has diffused out of the cell? Carbon dioxide. Only half of the energy that's being released is actually being captured as ATP. The rest of it is heat, a radiant form of energy. Okay? So those are the 4 final products: heat, water, carbon dioxide, and ATP. For the overall glucose catabolism, because carbon dioxide is only a product of the intermediate step and the citric acid cycle. Okay, but when we look at the overall process, it's one of our final products. Now, I briefly talked about brown fat in Bio430. Have the rest of you heard of brown fat? We have white fat and brown fat in the body. Brown fat looks different in electron micrograph. It also looks different in light microscope. It has individual lipid droplets rather than the entire cell being formed of a lipid droplet. But not only that, the mitochondrial processing is different. There's negligible amounts of ATP formed from glucose catabolism. Almost all of the energy is released as heat. So brown fat is very important in very young children and infants because of their high surface area to volume ratio. Heat is lost at a much faster rate. They have a higher metabolic rate to maintain that. So mice are going to have a higher metabolic rate than an elephant because of that higher surface area to volume ratio. And having the brown fat, in us it's limited to right around the heart area, whereas in very young children and infants it's kind of the trunk or torso area. And this provides an ability to keep up with their heat loss by not making ATP and releasing most of that as water.